So for the final talk of today, I'd like to introduce Dr. Rosa Mora from EMQN.
Thanks,
to you,
Carl.
Rosa.
Good afternoon, everybody. Today I would like to discuss with you what we can do to accelerate the implementation of point-of-care testing pharmacogenomic in clinical setting and what EMQN is doing to contribute to this field. First of all, let me introduce ourselves very briefly. We are a non-profit organization providing external quality assessment to laboratory performing diagnostic clinical tests. And we have more than 3,000 laboratories across 84 different countries which participate to our scheme. We can offer more than 60 EQA schemes, and every year we evaluate more than 20,000 reports with the help of 300 volunteers who are expertise in one of the areas associated with our scheme. Our aim is to ensure that every patient can access to genetic tests that are performing in a reliable, accurate, and comparable way. Let's now talk very briefly about pharmacogenomics. Pharmacogenomics is the study of the relationship between an individual genetic makeup and its response to a given drug. The number of gene variants that are associated with a drug response increases day by day. Indeed, I will be no surprise if every single of us will carry one of these pharmacogenomic biomarkers. And pharmacogenomics, therefore, can open the door to personalized medicine by pinpointing individuals who will have an unexpected reaction to a given drug or will respond in a different way. Most of the pharmacogenomic biomarkers are associated with a very common drug, and therefore pharmacogenomics also represent a cost benefit for the healthcare system. However, despite this benefit, pharmacogenomics is not really used in clinical setting yet, and one of the limitations is because the traditional genetic tests are performed in centralized laboratory, and this takes several days. Now, in most of the clinical scenario setting, such as acute setting, this time scale is not appropriate. So in a clinical scenario where a reactive approach is required, such as an acute setting where the therapy needs to be prescribed in a very fast turnover, an alternative to this traditional genomic test are point-of-care testing in genomic, which are technology able to identify a specific gene variant in a clinically relevant time frame. However, the development of such technology, which include the ability to perform DNA extraction, DNA amplification, and detection of the variation in the clinical relevant time frame, that sometimes is as short as 20 minutes, is not that easy. Indeed, currently, there are only few of point-of-care testing in genomics. And today I would like to discuss with you about the one that have been developed by a company called GeneDrive in Manchester with the collaboration of the NHS Trust. So the two point-of-care tests in genomics developed by this company are the mtRNA1-ID technology, which is associated with a clinical scenario of avoiding aminoglycosides-induced hearing loss. And the other one is the CPC219-ID technology, which is associated with a clinical scenario where optimization of antiplatelet therapy is required. Both these point-of-care testing in genomic have been recommended by NICE, and the RNA1-ID technology is currently used in 9 UK hospitals. So what it is, this technology? Is a qualitative in vitro molecular genetic testing able to pinpoint newborn at risk of hearing loss as a result of aminoglycosides treatment. How can it do that? By detecting a SNP in a mitochondrial gene, RNR1, which is known to be the cause of deafness after treatment, for example, with the gentamicin, and does so all in 26 minutes. For this reason, this point-of-care testing can be used in the neonatal intensive care unit, where the antibiotic treatment needs to be prescribed within an hour from when the baby are admitted to the unit. The other point-of-care genomic test developed by GeneDrive is the CP2C19 technology. In this case, the technology is able to predict a patient's response to drugs that are metabolized by the cytochrome P450, and therefore is associated with clinical scenario where, for example, clopidogrel need to be prescribed. Is able to do so by detecting SNPs in a specific allele of the gene CP2C19, which codified for a protein, which is involved in most of the metabolism of the common drug. In the table, you can see the allele that the technology is able to detect with the specific SNPs. And as you can see, most of these SNPs in this allele, they will result as a loss of function in terms of enzyme activity. Therefore, a patient carrying one of these SNP will either be an intermediate or a poor metabolized, and therefore will benefit by an alternative drug or a different dose. However, in these slides I am schematizing how both technology work. Basically, it's a very simple workflow where a BACO swab is taken by the patients and then is placed in a solution to allow DNA extraction, and then part of the solution is transferred to a sample cartridge where the lyophilized reagent for the amplification are present, and then the amplification is performed into the point-of-care testing devices, which is basically a thermocycle, which is able to detect the SNP by analyzing the melting curve. So as I mentioned, the mtRNA1 point-of-care test technology is currently used in 9 UK hospitals, and the reason is because it was successfully evaluated in a pilot study where these devices were implemented at the St. Mary Neonatal Intensive Care Unit setting in Manchester. And during this study, 424 babies admitted to the unit were tested with these devices, and 3 babies were actually identified with the variant, and therefore an alternative antibiotic to gentamicin was prescribed. The most important conclusion from this study was that actually this technology could be integrated in the intense unit setting without disrupting the normal clinical practice, which obviously is very important. Following the success of these pilot studies, another project called Devote has been funded by Innovate UK, and the aim of this project is to bring together academic, clinician, and industrial partner to facilitate the implementation of the current point-of-care testing available, such as the CP2C19 testing, but also to design, create, and implement new point-of-care testing in genomic. As for any medical devices, therefore also for point-of-care testing devices, the implementation test can be facilitated by participating to EQA schemes. However, at the moment, there are no EQA schemes available for point-of-care testing devices. That's the reason why EMQN is part of this Devote project and is collaborating with academic, clinician, and industrial partner to develop a EQA for the CP2C19 point-of-care testing device in genomic. We used the mtRNA1 as a case study because it's already in use. And basically, we, studying these point-of-care testing devices, we identify the best possible artificial material that can be used in the EQA schemes, which was identified as a stable cell-based swab. And the reason is because this artificial material need to be as close as a clinical sample, which in this case is a swab chicks. And also, of course, need to be stable and safe to use outside a laboratory setting. We use a proprietary method to create successful this kind of swap at different cell load, and we extensively validated the material to ensure intra- and inter-batch reproducibility and stability. Here I am schematizing the workflow that we used to design and optimize our EQA material for point-of-care testing in genomic. And so first of all, we have to establish cell line derived from patients bearing the mtRNA1 variant, and then we use these cells to manufacture swap that mimic the patient's cheek swap. And then we extensively evaluated this swap as EQA material for gene-drive mtRNA1 system. And by performing verification and validation with both quantitative and qualitative methodology that were developed in-house. And obviously the result of this verification were used to optimize the design of the swap until the result that we got from our internal validation were concordant with the one of the gene drive validation performed by using the patient's swap. So we are glad to say that we successfully optimized the design of the artificial material for point-of-care testing genomic. And we are actually producing not only verification package, which will allow the point-of-care team to assess their own service and also the quality of the devices that they are using, but we are also producing validation package, which will accelerate the implementation of the machine in the clinical setting. Indeed, at the moment, this is performed by using patient sample, and actually for each point-of-care testing in genomics, at least 40 patient samples are required before the machine can be officially be implemented. You can understand how, for example, in the case of the RNA1 point-of-care testing, where only 1 over 488 patients will have the mutation, this process may take a long time. So we will soon launch the pilot for the RNR1 point-of-care testing in genomic EQA scheme. And this is an overview of how the scheme will look like. So basically, participants will be provided with 3 different cell-based swap carrying the same cell load and the same gene variant. The point-of-care team will be able to use this swap as a normal patient sample, therefore they can be introduced in their normal clinical routine. Then the participant will return to us the result per devices, and then we will compare the result among all the participants, and we will give a feedback to highlight if any problem has been highlighted, and we aim to do this in a very short time frame. The mtRNA1 EQA scheme will be launched in September as a pilot scheme and will be followed by the CP2C19 early in 2025. So why, of course, the point-of-care team should use this EQA scheme is, first of all, to highlight problem with the kits and or devices and therefore ensure that the patient has the best care possible. But also they can access to continuous education and training, which in this area is particularly important since the test is performed by no specialized personnel. Also can facilitate the accreditation to the ISO. With this, I'm concluding. Of course, I would like to thank you, all the people that help us in providing this novel EQA scheme. So we have collaborated with the University of Manchester. We also collaborated with the Gene Drive team. And of course, I want to thank you all the EQA team that helped me in this journey. And thanks to all of you for listening to me. Thank you.